Welcome everybody to another one of our IPDGC Parkinson's disease webinar series. I'm really excited for this one. So my name is Natasha and I'm moderating this session. I'm a PhD student at the University of Luxembourg, where I work with midbrain organoid models to study the progression of Parkinson's. Today I have the absolute pleasure of moderating this talk from Professor Helene Plun-Favreau. And she's a professor at the UCL Queen's Square Institute of Neurology. I'm not going to go too much into her journey because I think she has a really nice slide to tell us actually about her career journey so far, so I'll let her do that. But just about the work that she's doing. So since Professor Plun-Favreau's arrival at UCL, her lab has carried out significant work on the molecular pathways associated with mitophagy and other mitochondrial dysfunctions in neurodegenerative conditions. In the last few years, her lab has specialized in developing high-content mitophagy screens in order to identify new Parkinson's and other neurodegenerative disease risk genes. So her lab uses a variety of different techniques and various different cell models, including patient iPSC-derived brain cells. And so, yeah, I'm super looking forward to hearing more about the work she's doing, about her journey. And with that, I would like to hand over to Professor Helene. And thank you so much for being here. I'm sure everyone is excited for this talk.
Thank you, Natasha. You can call me Helene. And I would like to say thank you to Fatuma as well, to the both of you for organizing the symposium today. So yes, so what I am going to discuss today is really mitophagy and Parkinson's disease, and more precisely what I am going to try to tell you about is how we've learned from the genetics of the Mendelian forms of Parkinson's disease to understand the biology of mitophagy, and how in return we can actually use or try to use this biology of the mitophagy to inform the genetics of the most common sporadic forms of Parkinson's disease. But really, first I was asked, as Natasha said, to give you a little bit, tell you a little bit about my background. And I am particularly happy to give this talk today for IPDGC-Africa. I was just telling Natasha and Fatuma before we started live that actually I have spent most of my childhood in Africa, and I've stayed in love with Africa ever since. So I was particularly keen to actually give a lecture in this symposium. So I was born actually in Paris, so not very original in France. And then when I was 2 years old, I actually moved with my parents, obviously. They were teachers in Burundi, so in Bujumbura in Burundi, where we stayed for a couple of years. And from there, we moved to Togo in Lomé, where I started my primary school. We then moved to Tunisia in Sousse, where I actually did another 2 years there before we go back to France for 4 years. So I started my secondary school in France, stayed there for 4 years, and then we liked Africa so much that we decided to go back to Africa, and this time we went back to Niamey, so in Niger. From there, I've done 2 years of my secondary school and then had to go back to France for my A-levels, and from my university, which I have done in between Angers and Poitiers, sorry, in France. I've then done my PhD in France, and after my PhD, I have decided to go for a postdoc in London, in UK, where I've been staying since. So a bit more about my research journey so far. So I have done my PhD on the signaling of the cytokines from the interleukin-6 family. So a bit of immunology, but I have really done mainly some cell signaling and some transduction. I finished my PhD, and then I was actually keen, I guess, to get a new challenge, but I was also keen to work in a more disease-related area. So I spent some spontaneous letter, all in the UK actually, and decided to do my postdoc with Professor Julian Downward at Cancer Research UK in London. During my postdoc, I was working on this protein, which is called HGI2, and that we knew was a protein which was located in the mitochondria, and that we thought as well was important for cancer. Well, it was. But during my research, I quickly realized that HTRA2 was interacting with PINK1, and I'll talk a lot about PINK1 during my lecture. It's a protein which is associated with autosomal recessive Parkinson's disease. And then really my research, when I was still at Cancer Research UK, my postdoctoral research has shifted from the cancer to the Parkinson's disease area. So at the end of my postdoc, I've decided really to stay in the Parkinson's disease, neurodegeneration area. And then I contacted Professor Nick Wood at UCL Queen Square Institute of Neurology, and I applied for a Medical Research Council career research development, which I was awarded. So I started my independent group at UCL at the Institute of Neurology in 2007. And since then, I've been really working on mitochondria still, but more particularly on mitochondrial quality control and mitophagy, and on neurodegeneration, and more particularly, although not only, on Parkinson's disease. So as you can see, it was not only in my lab, but I've been traveling a lot, but even here into my research, they have done quite a number of jumps. And I guess if there are some young researchers listening to me today, I think the piece of advice maybe that I could give is, at least in my experience, is it's good to have a research track, but I think it's also good to not be sad blinded and to be open to the opportunities that may open to you. I really had not planned my career as it happens now. And then actually, I'm really pleased I didn't, because I really discovered and I learned a lot on the way. And I think it's actually enriched my research to be able to work in different areas and not seeing everything just one way. So I joined in 2007. Then I was offered a senior lecturer position at UCL in 2013 and a professor position in 2019. Well, so, but that's enough really about me now. I think you guess it's more interesting to hear about my research, my science. But before I do that, I would really like to thank my group, really. Without them, of course, I wouldn't be talking today. And I can really say genuinely that I'm lucky enough to work with an amazing team. They're not only very talented, but they're also fun, they're very kind. And I suppose that working in a kind and supportive environment, especially when we are going through the tough times that we are going at the moment, is particularly important. So I'm very grateful to all of them. Today, you're going to hear more about the work of 3 people in the lab. First is Daniela Melantri, who is a last year PhD student in my lab. Then Dr. Mark Suter, who is a postdoc in my lab for now 8 years, although he's about to start his independent career now. And also from Ben O'Callaghan, who joined my lab only a few months ago, but who was already done an amazing job just the few last months that he was with us in the group. So the layout of my talk, what I'm going to try and do is just give you a little bit of background on mitophagy and Parkinson's disease, although I guess this audience might know quite well Parkinson's disease and the genetics of Parkinson's disease. Then I will try to tell you, as I said, how the genetics of the Mendelian forms of Parkinson's disease have actually informed us on the biology of the mitophagy. And finally, I will present some more recent work that we've done trying to using this biology of mitophagy to understand better the sporadic and most common forms of Parkinson's disease. So starting with the background on mitophagy and Parkinson's disease. So I guess you all know that the mitochondria are very important for a number of reasons, but in particular, they're very important for generating ATP or energy. And then it is perhaps not too surprising if they're so important. They are important in any cell type, but they're probably even more important in neurons because, of course, axonal transport, synaptic transmission, etc., cost a lot of energy to the neurons, so they need a lot of energy. So if they are so important, and in particular in neurons, it's not surprising that you have very elaborate mitochondrial quality control pathways. And you have really 3 main quality control pathways. The first one You see my mouse here is on the left and is really the mitochondrial chaperones and the mitochondrial proteases. So basically, vast majority of the mitochondrial protein are imported from the cytoplasm into the mitochondria. And once they are in the mitochondria, they need to be properly folded, hence the mitochondrial chaperones. Then you have some mitochondrial proteases, which are there in case some of the proteins have not been folded properly, for example, or some proteins are damaged, and in case they will be there to degrade these proteins, because the damaged proteins are known to be very toxic to the cells and to the neurons in particular. So you really have these mitochondrial chaperones and these mitochondrial proteins, but one first layer of mitochondrial quality control. Then you have a second pathway, which is a little bit maybe less known, but which has been well described by Heidi McBride and Ted Fon at the University of McGill in Canada, and which is the mitochondria-derived vesicles. So these mitochondria-derived vesicles, or MDVs, are containing some damaged proteins or components in the mitochondria, and they're able to bud off the mitochondria and to fuse with the lysosome for degradation. So that's the second pathway. In these two first pathways, you will have seen that only a subset of the proteins are damaged and needs to be degraded. Then sometimes what happens is that your mitochondria altogether is actually too damaged. And then if it's too damaged, then it needs to be eliminated altogether. And that's actually what mitophagy is about. So the mitophagy I'm going to talk about today is really the selective degradation of these damaged mitochondria, as opposed to a bulk and random degradation of any mitochondria you can have in the cells. So I don't have to discuss that today, but it's been thought, at least for a very long time, that this mitophagy is actually a cell autonomous process. So we think, for example, it happens in the neurons. It is suggested now here and there that actually it might be possible that the mitophagy might not really happen in the neurons, but rather in the surrounding glial cells, something which needs to be confirmed. But I just point that because I'll just show you one slide just now to tell you that there is an increasing role of the glial cells in mitochondrial quality control. Finally, I've just added apoptosis because, of course, if you have extensive mitochondrial damage, then that can just lead to the cell death altogether. So as I said, the glial cells play an important role in mitochondrial quality control, or so we think, actually, more and more. And I'm really not going to have time to talk about it today, but it has been increasingly recognized and shown that mitophagy, but also the mitochondria-derived vesicles, could actually play an important role in neuroinflammation. So I'm not going to have time at all to discuss these studies today, but really, if you were interested, I would encourage you to read the work of Rich Dule at NIH or Michel Desjardins at McGill University. Back to what the interest today, which is really the mitophagy, and as I said, the selective mitophagy. Here is basically how it works. And on all of my slides, I try to put the healthy mitochondria in blue and the damaged mitochondria in red. So what happens is that when a healthy mitochondria is going through a stress or a mitochondrial stress, it becomes partially damaged. And the mitochondria have a mitochondrial membrane potential. I don't want to go in any details here and confuse everyone, but maybe the only thing that you should remember is that the mitochondrial membrane potential is a key indicator of mitochondrial health. So basically, the more your mitochondria is healthy, the more polarized it is, and then the more damaged it is, then it becomes depolarized. So this partially damaged mitochondria, which has undergone a mitochondrial stress, is going to fuse. You might know as well that the mitochondria are actually dynamic and that they go through constant cycles of fusion and fission. So this partially damaged mitochondria, when it's actually going to fist, instead of giving two even daughter units, is going to give one mitochondria daughter unit, which is very healthy, very polarized, this one in blue. This one is actually going to fuse back with the healthy mitochondrial network and regenerate it. So really, this is very important here to maintain a healthy mitochondrial network. The damaged mitochondria, on the other hand, which is also very, as I said, depolarized, this one is going to generate a lot of reactive oxygen species. It's going to be very toxic and it needs to be degraded. Otherwise, it is very toxic for the cells, for the neurons again in particular. And so that's where mitophagy intervenes. So mitophagy is important for two reasons, really, two main reasons at least. It is, of course, to avoid the toxic accumulation of this heavily damaged mitochondria in red, but we often actually forget that it is also very important to maintain the healthy network of mitochondria on the other hand, and of course, maintain an optimal energy productions by the cells or by the neurons. So most of what we know about this mitophagy that I'm just going to talk to you about in a bit more details now is really coming from the Parkinson's disease genetics. And again, very briefly here, because I'm not sure I need to present Parkinson's disease to this audience, but you will know that it is a progressive neurodegenerative movement disorder affecting about 1% of the population above the age of 65 years old. You will know that it's characterized pathologically by the degeneration of the dopaminergic neurons and by the presence in the survival neurons of intracytoplasmic inclusion, which are called the Lewy bodies. The clinical features of Parkinson's disease are a tremor, a rigidity, a slowness in the movement, which is also called bradykinesia, and this characteristic here, postural instability, which was described by James Parkinson in his essay on the shaking palsy in 1817, I think that was, so quite a while ago now. More important maybe for message for this lecture, what we know as well is that the toxins that can actually poison the mitochondria can induce Parkinson's disease, and we know that since the beginning of the 80s now, so quite some time, really suggesting again that mitochondria play an important role in Parkinson's disease and mitochondrial dysfunction. And what we also know, as you will know, is that if most of the cases, 90–95% have no known genetic cause, 5 to 10% are actually familial cases of Parkinson's disease. And here is actually what the architecture of the Parkinson's disease genetics look like. And again, I'm sure you will have seen these slides a number of times, but you have three main groups of genes. The first group of genes here are actually in the top left corner, are actually the rare Mendelian genes, which are of high penetrance and high effect. These genes can be of can have an autosomal recessive or autosomal dominant mode of inheritance. Then you have this group of genes in the middle, which we would call the strong risk factors, and for which the penetrance is not complete. And finally, you have this last group of genes here on the bottom right corner, which really one could call maybe weaker risk factors, meaning that these genes are generally low risk, but high frequency genes. And all these genes have been identified by GWAS, the Genome-Wide Association studies. I'll say a word about them a bit later. But first, as I said in the introduction, I go back to my mitophagy, and as I told you, most of what we know from the mitophagy biology is really coming from the Parkinson's disease genetic, and is coming more particularly from two genes associated, two Mendelian genes associated with autosomal recessive Parkinson's disease, PINK1 and Parkin. So I make a very long story short, and I told you how PINK1 and Parkin together can actually orchestrate the mitophagy process. So PINK1, to start with, is a mitochondrial protein, a mitochondrial kinase. So it's kinase which is imported into the mitochondria. And usually in this healthy mitochondria, blue mitochondria, as I told you, which have a normal mitochondrial membrane potential, PINK1 is imported into the mitochondria and is very quickly degraded. So what happens is that actually you usually hardly can detect it. It has a very high turnover in basal condition. On the other hand, if your mitochondria become damaged and therefore depolarized, then they can't be imported into the mitochondria, and instead PINK1 accumulates at the surface of the mitochondria. So that's what you would see here by Western blot. Very simply, if you take a mitochondrial fraction, so here is a mitochondrial marker, the complex 5 at the bottom, and here you have untreated cells, and here treated cells with CCCP. It's actually already a barbaric name, but what the CCCP does is that is actually very brutally depolarizes your mitochondria and mimics, if you'd like, a damaged mitochondria. And what you see, as I told you, is that in basal condition, you can't detect PINK1, the turnover is too high. On the other hand, if you depolarize the mitochondria, PINK1 can't be imported and degraded, and as a result, it accumulates, if I'm trying to get my mouse, it accumulates here at the surface of the mitochondria. When PINK1 has accumulated at the surface of the mitochondria, what happens is that it then recruits Parkin. What we see is that we don't know exactly how that happens, but what we know is that this PINK1 accumulation is necessary for Parkin to be recruited. Parkin is another gene associated with autosomal recessive Parkinson's disease, and it is an E3 ubiquitin ligase. So it's tagging with ubiquitin the protein that are targeted for degradation. So PINK1 accumulates, then Parkin recruits to the mitochondria. Once it's at the mitochondria, as I told you, so sorry, first of all, you can see the Parkin accumulating here. You can see it again by Western blot here. Same thing in your mitochondrial fraction. PINK1 accumulates, that recruits Parkin into the mitochondrial fraction. You can also see that by immunofluorescence. In red here, you have the mitochondria, which are stained, and in green, these puncta are actually Parkin. I didn't put a picture here, but in basal condition, Parkin would be everywhere in the cytosol. When only you depolarize the mitochondria and you induce mitophagy, you see this puncta of Parkin, this green puncta appearing, and this puncta perfectly localized with the mitochondria. So now Parkin is at the mitochondria. As I told you, it's an E3, so ubiquitin ligase. And what it does is that it's going to basically be redecorating the outer mitochondrial membrane with ubiquitin molecules that I've put in gray here. And now these ubiquitin molecules at the surface of the mitochondria, they're going to be like an eat-me signal for both the autophagy and the ubiquitin proteasome system machinery. So which I put here. And basically, thanks to the autophagy and the UPS, this damaged mitochondria, which is now redecorated with ubiquitin molecule, is going to be degraded. And again, that's going to avoid, as I said, the accumulation of these mitochondria that are heavily toxic for the cells and for the neurons in particular. Now, it is really how we were assessing mitophagy. We still do, but it's really the basic methods. All of that is in lysed cells. What you can do as well is obviously use the mitochima. And what the mitochima is that it's a fluorescent protein that has a different excitation peak according to the pH. In other words, and in simple words, say that your mitochondria in a neutral or in normal condition at a neutral pH will fluorescence green. When the mitochondria fuse with the lysosome, so with the autophagy machinery, as you will know, the lysosomal content is very acidic, then the mitochondria are going to be turning red. And so by measuring this ratio between red and green mitochondria, you can know the amount, say, or the level of mitophagy of the mitochondria that are actually being degraded into the lysosomes. So I will show you an example later, but it's very simple. These methods I'm just showing you, mitophagy, biochemistry, or immunofluorescence methods, all of this and a large amount of the work, it's a flow in the field, I have to say, have been performed in cell lines as opposed to being performed in neurons, or even better in vivo. Measuring mitophagy in neurons, measuring mitophagy in vivo is another level of complication. And again, I'm not going to discuss it today, but happy to answer questions later if you would have any. I just really show you the basic methods that we've, so that you can understand the experiments that we've done later. So that was just a little bit of background on mitophagy and how, again, PINK1 and Parkin have informed the biology. What I didn't say, of course, is that the loss of function mutation in the patients with autosomal recessive Parkinson's disease in PINK1 and Parkin can actually lead to, or that's why we think, deficient mitophagy. By that I mean, for example, accumulation of these toxic mitochondria into their neurons, which we think is actually contributing, of course, to the pathogenesis of the disease. Another example of how you can learn from the Mendelian Parkinson's disease comes from my group. Sorry, but I'm going to be quick because it's a while ago we published that. And I give you this example of EBOX7. EBOX7 very quickly is another gene which has been shown to be associated with autosomal recessive Parkinsonism, and which is part of, of the SCF E3 ubiquitin ligases. Sorry. So also, again, a protein which is involved, like Parkin, if you'd like, in protein ubiquitination. And what we've shown in the lab a number of years ago is that, like PINK1 and like Parkin, FBOX7 also plays a role in mitophagy. We've shown that in different ways, but I just show you 2 free experiments to give you an idea. First of all, what we've shown in cell line to start with is that actually, again, if you depolarize your mitochondria, what you see is that with time, FBXO7, which was in the cytosol, is slowly depleted from the cytosol and is slowly instead relocating to the mitochondria. What we've also shown, and using cells, fibroblasts in that case, from patients carrying mutations, from Parkinson's disease patients carrying a mutation in FBOX7, is that the patient line which is carrying this mutation in FBOX7 is actually deficient in ubiquitinating the protein I was telling you of the outer mitochondrial membrane. And finally, what you've done is try, as I said, to work in vivo. And again, it would be for a debate, but the PINK1 or Parkin knockout mice, or in fact, I think I can say safely, all the genetic models of Parkinson's disease have not been particularly useful in that respect, that they don't recapitulate any of the clinical features of the disease. I'm thinking none of them has any dopaminergic neuron loss or any cytoplasmic inclusion equivalent to the Lewy bodies, etc. The flies, on the other hand, they're flies and obviously come with limitations as well, but have been very useful to understand the biology of autosomal recessive, in particular Parkinson's disease. And what we have shown here in collaboration with Alex Whitworth at the University of Cambridge at the time is that actually, if you take the Parkin knockout flies, they have a number of deficiencies. Their mitochondria are deficient. That's what you can see in electron microscopy here. I'm not going into detail, but they also have problems flying and climbing, which would be the equivalent of a movement disorder, if you like. And they also have a small but significant loss of their dopaminergic neurons. And what you do is that you take these Parkin knockout flies. Of course, your control is to try to rescue all these phenotypes by overexpressing Parkin in the Parkin knockout flies. And you can do that here on the right, but here also you can see the mitochondria are completely restored. But quite interestingly, you can, that's Parkin here, sorry, that's the other way, but it's the same. But what you notice is that you can not only rescue the Parkin knockout flies by overexpressing Parkin, but also by overexpressing FBOX7. And what that does is that actually that again gives you a strong indication that what you see in the petri dish, in the cells, might also be true in vivo. And by that, I mean that FBOX7 and Parkin are actually working together. Right, now I go to really more recent work, and I will finish, as I said. Now, I hope I have shown you actually how we could understand the biology of mitophagy with PINK1, with Parkin, but also with FBOX7, so through the Mendelian form of the disease. But now what I would like to discuss is how, now that we know a bit better this biology, how can we use this biology to maybe develop some screening assays and then understand better the sporadic most common form of the disease. And the genes really we want to understand this time are really this group here in green that I was telling you about, which are really the weaker risk factors, as I said, that they are low risk but high frequency genes. And as I said, these genes have been identified by genome-wide association studies. Again, I think probably quite a few geneticists in the audience. I am not, but just for those who don't know, what the genome-wide association studies are doing in simple words is that they are searching the genome for small variations, which are called SNPs for single nucleotide polymorphism. So they're searching the genomes for this small variation, for the SNPs, and then actually for SNPs which are more frequent in the disease population, so in the population of Parkinson's disease patient with no genetic cause this time, or at least no Mendelian genetic cause, and try to understand them. So more frequent in the disease than they are, of course, in the control patient. So I give you an example here of the last GWAS study for Parkinson's disease, which was performed by the International Parkinson Disease Genetic Consortium, but which really was led by Mike Knowles and Andy Singleton at the NIH. And this last study, so which was published now nearly a year ago, has actually identified 90 most significant, if you like, SNPs, of these SNPs. 90 significant signals. And what a signal identified, unfortunately, it doesn't really identify a gene, but it identifies a locus. Each signal or most significant SNP identify a locus. I gave you an example here, and I picked the chromosome 16, not completely randomly. You'll see why in a second. And here, you know, I don't know whether you can see, but here I try to show with my mouse. In purple, you really have the most significant SNP, and then you see actually you have a peak. But then now let's look and see all the genes you can see that are under this signal. And then the question is, how do you do to actually know which are the genes? We know that this locus is important, but how do we decipher which of these genes, or genes, actually it could be several of them, are actually important, are increasing the risk of Parkinson's disease? For very few of these variants, I have to say, not many, but for very few of these variants, the gene has been identified, because, for example, on some of the SNP, the gene you have in the locus, you have alpha-synuclein, GBA, or LARC2, which we already know are actually either Mendelian genes or strong risk factors for Parkinson's disease. So that's okay. But that's only very, very few of these variants that have been unambiguously identified, associated with the genes. In the other cases, like this one, we really don't know. That's one example of a locus. I give you another example of a locus, again, which will make sense in a few minutes. And that's another locus, which is the second most significant locus in the Parkinson's disease latest GWAS, on chromosome 17. Again, you will see here, it's even difficult to find a peak at all. So try to understand which gene is the important gene is actually difficult. In that case, and I'll come back to it, you have tau in this locus. And then a lot of scientists have assumed that actually tau might be the responsible gene for Parkinson's disease, even though there is not a huge amount of evidence, or actually little evidence, I think I can say even, for any tau pathology in Parkinson's disease. So that's always been actually a question in the field. Is tau the responsible gene? Is it another gene on the locus? We don't know. And I'll come back to that. But here, all I wanted to tell you is that. So we have identified the latest GWAS 90 loci. We have this loci. And now how do we actually know which genes are important? And that's exactly the reason why, together with John Hardy at UCL, with Patrick Lewis at the Royal Veterinary College, and Alex Whitworth, the fly geneticist I was telling you about before at the University of Cambridge, we have been awarded this MRC program grant with exactly that aim. And the aim of this grant was the following. First of all, so you have 90 loci. Then, as I said, a number of genes under this loci, say you have at least 1,000 genes all in total. So the first thing we thought we do is that we are actually going to use genetics bioinformatics to prioritize the genes that we think might be the genes, the risk factors for Parkinson's disease in this loci. So we use bioinformatics and genetics first. Then in the meantime, and that was our job with my group, was to develop some assays for some pathways that we know are important for the pathology of Parkinson's, for the pathophysiology of Parkinson's disease. And of course, you won't be surprised to hear that the assay we have decided to develop is a mitophagy assay. So when we say we prioritize the genes, we develop some assays where we can do high content screening. Then we knock down using siRNA these genes one by one. And if actually, say, I pick a gene that I had prioritized, which I see play roles in mitophagy, it will give me much greater confidence that this gene is actually the risk gene for Parkinson's disease under the loci. So that's exactly what we've done. And for that, we've developed a mitophagy assay. And what I didn't say before to not overcomplicate it thing, but I'm saying you now only, is that we know that actually one of the main targets of PINK1 is actually ubiquitin itself. Ubiquitin was not even known before to actually be phosphorylated. Now we know that it is phosphorylated by PINK1. And what we know is that ubiquitin, when it is phosphorylated at serine 65, is an important marker for mitophagy. So here is what you see, for example, by immunofluorescence. So here you are in control condition, and in red you have your mitochondria. TOM20 is a protein of the outer mitochondrial membrane, so at the surface of the mitochondria. Then, of course, here you have your phosphoubiquitin signal, should be green. In basal condition, there is no phosphorylation of ubiquitin. Now, if we induce the mitophagy, again, this time you don't use CCCP, as I said, but the same thing. What you do is that you depolarize your mitochondria, you mimic damaged mitochondria, you induce mitophagy. And then what you do what you see that, first of all, obviously, ubiquitin starts to be phosphorylated at CERN 65. You also see, you detect in the red signal that your red signal starts decreasing, and the reason for that is that your mitochondria already start being degraded because the mitophagy has been induced. And then if you do exactly the same experiment in pink1 knockdown cells with a good knockdown, so remember pink1 is meant to phosphorylate ubiquitin, then what you see is that you lose pretty much all your phosphorylation of ubiquitin, and as a result, no mitophagy or nearly no mitophagy is occurring. So we've used this assay, all the way I'm going to talk to you about the phosphorylation of CERN65 or ubiquitin or phosphorylated ubiquitin as a marker again of mitophagy. And we have developed this high content screen. I won't go into details, but it has been a lot of work, and really thanks to Daniela in my lab, but we've also to the group led by Paul Whiting at the UCL Alzheimer's Research UK Drug Discovery Institute. So we have developed a high content screening assay. And then, as I said, in the meantime, we have prioritized the genes on the loci. We didn't work at the time with the latest GWAS, but with the previous one from 2017. But anyway, so it was not 90 loci, but still quite a number, and we prioritized 40 genes. I said we, it's actually completely untrue, because I really didn't do any of this. I am a cell biologist myself. This work has really been performed mainly by Claudia Manzoni, who is now a lecturer at UCL School of Pharmacy, and Dania Trabzuni, who was working with John Hardy at UCL. And what we've done is that they've actually used a colloq analysis. So what colloq analysis are doing is that they're using linkage disequilibrium EQTL information derived from two expression databases, which are Brainiac and GTEx. Then Claudia Manzoni had actually created a protein-protein interaction network. We know that actually if the proteins play a role in the same pathway, they're probably obviously have a chance to interact with each other, directly or indirectly. We've obviously looked at the GWAS, as I said, the previous one from 2017. We've included few Mendelian genes for control, and we have also included lysosomal genes, because it was shown previously that lysosomal genes are enriched in Parkinson's disease. So we've selected 40 genes only, purely for technical reason. It was easy in duplicate on a 96-well plate. Otherwise, we would have selected more, and I think we are actually going to repeat this screen with more genes now. But we've selected these 40 genes, and then we went on the screening. And here is what we found. So here, try to make this, but what you see again is that here, if you actually induce mitophagy by depolarizing the mitochondria, you see that actually, you see here, hop on the y-axis, that basically it is the phosphorylation of ubiquitin. Okay, so in non-stimulation condition, everything would be very basically at zero. Then you induce mitophagy, ubiquitin is phosphorylated. But you see is that that doesn't happen, as I've shown you, if you knock down pink1, but that also doesn't happen if you knock down this gene that we had prioritized, one of the 40 genes, which is called cat8. And I'll come back to that in a second. Let's skip that just for the sake of time. cat8, and I had said I had not picked this locus randomly, is actually one of the genes on this locus on the chromosome 16. And interestingly, maybe of our interest to some of you, it has also been suggested to be a risk factor in the latest genome-wide association study for Alzheimer's disease. So then we validated cat8. I go quickly because otherwise I speak a lot, and then but basically we do the immunofluorescence. We confirm that, like pink1, if you knock down pink1, you decrease obviously phosphorylation of ubiquitin. Same if you knock down cat8, although not as strong as a pink1 knockdown, I have to say. You can also validate that, Marcus and this Western blot, basically by biochemistry, where actually what you see here, this smear is actually, when you activate the mitophagy, is a smear for phosphorylation of ubiquitin. Again, with time, you can see an increase in the phosphorylation of ubiquitin. Scramble is your control, so in your control knockdown. If you knock down PINK1, you pretty much lose, again, same thing as the microscopy picture, you lose the phosphorylation of ubiquitin, and it's usually decreased as well in the catate knockdown. So, okay, we're happy we've confirmed our screen that actually that is the case. If I knock down catate, there is less phosphorylation of ubiquitin. So what is catate? Catate is actually a lysine acetyltransferase, and it's responsible for acetylating the lysine 16 on histone 4. Quite interestingly, although I have also, to be honest, we didn't manage to repeat this data so far, I think it is one of the only lysine acetyltransferase that has been shown, or at least suggested, to be located at the mitochondria in this Cell paper in 2016. So you have about 20, I can't remember, but it's about, say, 20 lysine acetyltransferase or CATs. So what we've decided to do is to knock down again each of these CATs one by one to see whether what we were seeing, the effect on phosphorylation of ubiquitin, was only with catate or basically was with all the CATs. And to make, again, long story short, what happened is that the effect that we've seen was only with catate, so very specific to catate and not to any of the other lysine acetyltransferase of the same family. So quite specific. But maybe more interestingly, catate, which is also called MOF, that said, which is here, is actually part of a big complex, which is the NSL complex, stating for non-specific lethal, and is a master regulator of transcription. So we said, okay, let's see, basically, we know that if we knock down catate or MOF, we decrease the phosphorylation of ubiquitin. What happens if we actually knock down the other components of the complex? And that's what we've done. And what we've seen is that in particular, if you're knocking down, obviously, catate, you see less phosphorylation of ubiquitin, but you see even a stronger effect if you knock down these other components of the complex, CANZL1, CANZL2, and CANZL3. And what was very interesting to us is, in particular, is CANZL1, because CANZL1 is on the tau locus that I was just telling you about, this chromosome 17. And I was telling you that a lot of people are actually, how should I say, assumed that tau is the risk gene for Parkinson's disease on this locus. Well, at least our data suggests that maybe it is tau, but at least maybe also CANZL1, maybe another risk factor for Parkinson's disease. Is responsible from a neurological syndrome, but again, I'll be quick there. So the same as but we validated the CANZL1 effect, like we've done with catate. So we've done that by immunofluorescence and by biochemistry. And in fact, the effect of the CANZL1 knockdown is even much stronger than the effect of the catate knockdown. It is nearly as strong as the PINK1 knockdown effect. And you can see that here in really this, if you do kinetic, so here what you see is basically a kinetic of stimulation with oligomycin, antimycin, so to depolarize the mitochondria. And then what you can see is that with time in black, so you have the control situation. You can see that in pink, it's always the same, but is obviously the PINK1 knockdown, where you have very low phosphorylation of PINK1. And each time, you know, catate and CANZL1 sit comfortably between PINK1 and the control, with every time CANZL1 knockdown having a stronger effect than catate. We also looked in, so everything I didn't say, but was in Parkin overexpressing cells, which is another possibly flow of the field, I have to say. A lot of these experiments, as I had said, are performed in cell lines, but also a lot of them in cell lines overexpressing Parkin. So we decided to repeat these experiments. Daniela has repeated them in the wild type cell line, which are not overexpressing Parkin, and we see the same thing, exactly the same thing as in the overexpressing cells. The next step, and that's the beginning, is that actually at the moment, Daniela is doing a lot of work to try to now optimize the CRISPR interference iPS-derived neurons models, in which we would be able, obviously, to knock down catate and CANZL1 and see whether we can confirm our phenotype, not in a cell line, but in a human neuron, of course. So that's going to be the next step that we are trying to do at the moment. I'm going to skip the Parkin accumulation because I sent none. The last thing I'm going to show here just for the biology is that I told you at the beginning that all I've shown you before just now is really in fixed cells. So we wanted to know really the real essay for mitophagy is to use this mitochondrial. So you remember this mitochondrial fluorescent protein, which is basically fluorescing green when the mitochondria are in the cytoplasm in a neutral pH, and that they become green where they're actually fused with the lysosome. So we've used this mitochondria, and here I make it short, but again, it confirms exactly what we've seen before. So you've seen the mitophagy occurring here in the control cells in gray, and then you can see all these three, so the green, blue, and pink are for cancel 1, cat8, and pink 1 knockdown cells. Again, you can see that if you knock down these genes, then you have less mitophagy in these cells. We have also done a little bit of work in flies with Alex Whitworth, and all we could see, we didn't really observe anything in the cat8 flies, but in the cancel 1 knockout flies, or the equivalent in flies is NSL1, is actually that we could observe some neurodegeneration in these flies. I told you actually that cancel 1 is located within this the tau locus, so which is basically an inversion polymorphism, and that this inversion polymorphism is also containing tau. And tau has been obviously, how could I say, postulated by many to actually be the risk gene at this locus. The thing is that now it is very, very difficult to determine whether it is tau. It is a very complex genetic region. I'm not a geneticist myself. It's still for me very difficult to get my head around it. But basically with the H1, H2 haplotype, it gets very difficult. You start having some haplotype specific SNPs, and it's difficult to study this region. So what Andy Singleton is going to do at the NIH or the IPDGC is trying to do long gene sequencing to try to again decipher whether tau might be the gene, whether cancel 1 might be the gene, whether two of them might be the gene, whether there might be other risk genes important in this locus. All we can do is actually again use our screen, and what we've done is that we've taken all the genes under this locus. They're all listed here. Actually, I can't remember, but maybe again 20 or 30 genes under this locus. And again, the only gene, the only knockdown we see an effect with on mitophagy is cancel 1. Again, I am not trying to claim that only cancel 1 is the risk gene on this locus. We're only looking here at phosphosense 65 as a proxy for mitophagy. And of course, tau might be important as well, and might be important in other pathways involved in Parkinson's disease. So really what I've been showing you here is that cat8 and cancel 1 are two new Parkinson's disease G1 hits, and that they both interact. I didn't say, but it was shown before, and that in the NSL complex, obviously, that they might be, I should say, partially located at the mitochondria or relocated at the mitochondria, and that we have shown that they are both implicated in mitophagy. Now, the thing we really wanted to know is about the mechanism, really. And I have only one slide, but Ben, in the few months he's been here, has done a lot of work and actually has shown, as I told you, the NSL complex, for which both cat8 and cancel 1 are part of, is a master regulator transcription. And what Ben has shown is that actually, if you knock down cat8 or you knock down cancel 1, more specifically cancel 1, actually you decrease the transcription of PINK1. So you decrease the PINK1 mRNA level, which of course would explain if you have less PINK1 mRNA, less PINK1 protein, then you have less mitophagy. So cat8 and cancel 1 regulate the PINK1 mRNA level. I didn't go into the details into the genetics data, really, because it's more the work of the group of John Hardy and Mina Heighton here. But what they've shown is that the genetics data are corroborating our functional data. And by that, I mean is that if I look at EQTL, we see that actually, or it is predicted, the genetics is predicting that less of these genes, which actually increase the risk for Parkinson's disease. And that's what we see in our screen. If you knock down this gene, then the mitophagy is deficient, and we know the mitophagy is protective. I didn't go into details either, but what is quite striking is that usually when you look at SNP, in a vast majority of cases, the SNPs are in uncoding region. In our case, 2 of the SNPs are actually in the coding region of CANZEL1 and are actually missense mutation in the CANZEL1 genes. And obviously, we are going to study these variants in the future, of course. What I think, or what we think it is important, is that because so far the mitophagy was only shown to be associated really with the rare Mendelian forms of Parkinson's disease, and that now our work is suggesting that it might also be important for the most common sporadic forms of disease, but also because actually nobody has investigated the possibility that regulating lysine acetylation may be a good way of regulating mitophagy, and that's some work we are going to pursue when we understand the function of K8 and CANZEL1 better with the Drug Discovery Institute at UCL. That is it, and just to tell you that mitophagy has since also been shown to be important for other neurodegenerative diseases, and in particular frontotemporal dementia. So again, you show that when you understand really better the biology of a pathway, how it can be informative for other areas and other diseases. And I really want to finish again by thanking my group. Emily and Bea are not on the pictures here, but obviously that was a picture here of us having a picnic this summer. And again, I've highlighted here Daniela, Ben and Mark, who have actually performed a vast majority of the work that I have presented today. I will finish by thanking also my collaborators, in particular John Hardy and Dania at UCL, but also the group of Mina Reiten and Patrick Lewis at the RVC, Claudia Manzoni at UCL, and finally the group of Paul Whiting at the DDI. Thank you very much, and apologies for going a bit over time. Sorry, Natasha.
No problem. Thank you so much, Helene, for that. It was a very interesting talk. I think you touched on a lot of very interesting aspects related to mitophagy and PD in general. So there's a couple of questions in the chat, and I also have a few questions. So there's one question coming from Aaron. So Aaron from Congo. And he was asking, so he says that, sorry, so histone methyltransferases have been reported to have an effect on mitophagy. Are they involved in the regulation, I guess, of Parkin or PINK1? Do you know anything about that?
No, that's something that actually Ben has really started to look into, but that we would need to go in more details. We are really at the very beginning of investigating the mechanism. I didn't go into details. We've seen this effect on transcription, but and of course it could be through lysine acetylation. But I suspect PINK1 and K8 might also have other effects on the mitophagy pathway.
Okay, thank you. Another question that came in, which I also had, so since in Parkinson's disease, the accumulation of alpha-synuclein is one of the hallmarks. Have you speculated or theorized how mitophagy could be linked with this aggregation of alpha-synuclein and the occurrence of Lewy bodies?
No, it's a good question. And you have actually few studies, not that many, but few studies that are actually suggesting. I'm thinking of a study in particular from the group of Christian Haas, which is suggesting that actually alpha-synuclein might also play a role in this pathway in mitophagy. But otherwise, a large majority of the work on alpha-synuclein has more been associated with either bulk autophagy or with chaperone-mediated autophagy, as opposed to this selective mitophagy. So I would say that at the moment, the link between this selective mitophagy and alpha-synuclein accumulation is
it's not clear.
Yeah.
I'm just wondering, are there any shared parts of the mitophagy and autophagy pathway? Because I guess there is some similarities in the pathway. So maybe there's some proteins that are shared within the two pathways that might be influencing.
Yeah, of course, it is a very good question. In fact, there is. So what it happens is that some of the components are shared between the bulk autophagy and the selective mitophagy. And one example is, of course, for example, LC3, I'm thinking, mitophagy. Some other components are actually not shared. Another component which is shared is P62, although there has been a lot of controversy as to whether P62 is really required for mitophagy to occur. And in fact, more of the evidence, and we have a paper coming out actually showing that at least in P62 CRISPR knockout iPSC neurons, we don't see any effect of the P62 knockout on PINK1 mitophagy. But you have some components which are shared, some components which are not shared. So that's a good question. Yeah.
Okay, super interesting. Another question that I had, you briefly touched on this, but I found it quite interesting, this idea of transmitophagy. So I don't know if you could just explain a little bit more. So from my understanding, it's that the mitochondria from the neurons would actually be degraded, so to speak, by glial cells and not within the neurons themselves. Did I understand that correctly? And what's the implication of that if it is occurring?
Yes, so it's only a few papers, but I think they are very interesting, even though there is little evidence. But I have to be honest, I like the idea more than anything else. What this maybe couple of papers are suggesting is that basically, when there would be some damaged mitochondria in the neuron, they would actually be able to get rid of them, to unload them. And in fact, they have some super nice TEM showing even, like you see, like the damaged mitochondria budding off the axons. That could be, I don't know. Again, it needs to be confirmed, but this paper is nice. And then what they suggest is that on the other hand, these damaged mitochondria are unloaded by the neurons and taken up by the glial cells, which can then degrade them. And that makes sense because I didn't go into details, but I said the neurons are very dependent on mitochondria for generating energy, but they are even more so than other cell types, such as glial cells, which are more actually, which have higher glycolysis and don't probably need as much their mitochondria for generating this energy. So these models are actually suggesting that, the neurons themselves could unload their mitochondria and then they would be degraded by the glial cells. And you have a paper in Nature maybe 2 or 3 years ago, and I can't remember the group now on the top of my head, but which is suggesting that on the other hand, what happens is that during an hypoxic situation in particular, the glial cell would be able to fit the neurons back with healthy mitochondria. So you would have this exchange wherever the neurons can't degrade the damaged mitochondria, but then they send them to the neurons and the glial cells, and the glial cells can actually send back or give back to the neurons with healthy mitochondria. It's very, the idea is very, very attractive. I have to say, and that's something I would have liked to work on in a long time, but I think it still needs to be proven.
Yeah, absolutely. It's a very interesting
the thing which is, I should say as well, and that we've done, is that if you compare the rates of mitophagy in iPS astrocytes, for example, as compared to iPS neurons, it is nothing to compare actually. The mitophagy takes a long time, 1 to 2 days to be induced in the neurons. And again, probably because they need the mitochondria so much for generating energy that they probably have some kind of blocking mechanisms, not lose their mitochondria. On the other hand, if you try to induce mitophagy in astrocytes in a few hours, in 1 to 2, 3 hours, it starts. So definitely a lot more mitophagy in the cells which are more glycolytic.
It makes sense. It's a really interesting hypothesis. I hope more comes out on
this
particular topic.
It was a few years ago and not as much. I still like the idea. I still present it, I think,
yeah, it's interesting.
More for the discussion, but I think it still needs to be confirmed.
No, absolutely. Another question from Dr. Pawan Singh. What are your thoughts on PINK1's role besides mitophagy?
Yeah, but the role I didn't have time to discuss it, but I think the role of neuroinflammation again is very interesting. Although again, it's really, as I said, coming from mainly 2 groups, and I suppose there is a lot more to come. But at least it would make sense to me that, for example, mitochondrial antigen presentation or whatever is important in neuroinflammation. But that's something that we've not been investigated ourselves in the lab, and so I can't really comment further, at least in our own hands. But quite a few papers coming out. If you are interested, sorry, I can't remember your name, but check the latest paper from Rich Duhl, as I said, or Michel Desjardins, and then you will see.
Okay, thank you. And okay, 6. I guess the last last more comment from me than a question. I got quite excited towards the end of your presentation because coincidentally during my master's I was working on Cancer 1.
Oh, really?
Yeah, in iPS-derived neurons. So we're trying to establish a
Who did you work with in the Netherlands or
in the Netherlands? Yes.
With Neil?
Yes, exactly.
We're collaborating with Neil. Actually, he's about to send us the cells and everything.
Okay, exactly. So I was there when they kind of started this project
really?
three or 4 years ago now, exactly.
That's
so when
really
I heard you talking about that, I started making the association between Cancer 1 and then now realizing that it's also associated with PD, and now I'm working on PD. I found that link to be super interesting.
Oh, that is exactly, yes. We've been awarded this grant by the Michael J. Fox and Ben, and they've been talking about us actually the postdoc on this grant, and on this grant we are going to be collaborating with Neil. He came, invited him to give a talk as well at UCL, like just before the pandemic.
Oh, okay. No, that's pretty cool. So really exciting work. Okay, so on that note, I don't see any more questions. So I'd like to thank you again so much, Helene. I really enjoyed this talk.
Everyone.
Thank you so much. And thank you everyone for joining today.
Thanks to Fatuma again. Thank you, Natasha.
Thank you. Join us again for the next talk in a month, at the end of the month, every last Tuesday for these talks. And we will see you then. Thank you.